Good morning, dear children. Last class, we discussed about monohybrid cross. And you know the scientist who had done this monohybrid cross. Who is that? Yes, Gregor Johann Mendel. And you know that why he did this cross also. For what? To find out what? Yes, how the characteristics or characters are transmitted from one generation to another. That means why these offsprings or why the children are similar to the parent. Not only that, why there is differences also. So he wanted to find out. For that, he tried to do the experiment in which plant? Garden pea plant. And who can say why he selected garden pea plant? You know that in the garden, different types of plants are there. Rose plant, jasmine, lily and all. So why he selected the garden pea plant for doing his experiment? Yes. Some reasons are there. First one, it's easily distinguishable characteristics. Very easily he could find out, differentiate one character from another. And very easy to grow. And short lifespan. And the flowers are bisexual. So these are some of the reasons that helped Mendel to select the garden pea plant for doing his experiments. And as I told you, the main reason he selected for doing his experiment is because of the 7 easily distinguishable characteristics. Male 5-7, you know that when we observed that he could see the height of the plant. Some plants are tall and some plants are dwarf or short. And the color of the flower, purple. Next one we could see white. Purple color flowers he had seen and white color flower. Purple, otherwise we can say it is violet, purple or white. Then the shape of the seed, that is round. Next one, it is the wrinkled. Round, so 3 characters over. Then the shape of the pod, it is the inflated. Shape of the pod, inflated. Next one, it is the constricted. Color of the seed, green, yellow. Shape of the color of the pod, that is yellow, green. And position of the flower, that is axial and terminal. So these are the different or 7 contrasting characters that helped or that made Mendel to select the garden pea plant for doing his experiment. So we had already done, and this I know these all are the dominant. And among these 2 contrasting characters, one character is always dominant and one character it is always recessive. One character is always dominant. And how he selected or how he reached a conclusion that only one of these characters, if he considered tallness and dwarfness, tall is the dominant one. So how he reached that conclusion? After doing a lot of experiments in the garden pea plant, he reached the conclusion that some of the characters of this garden pea plant are always dominant. That means they appear in the first generation. When he crossed 2 plants, in the first generation, that is one is father and one is mother. When he crossed these 2 plants in the first generation, he always got one answer or one character expressed. One character expressed. For example, he crossed the tallness and the dwarfness. In the first generation, he got all the plants as which size? Tall. So after doing like this so many experiments, you know the flower color purple and white. Purple color is the dominant one. And you know the shape of the seed, round and wrinkled. Round is the dominant and wrinkled is the recessive. So likewise among these characteristics, one is dominant and another one is recessive. So after doing lot of experiments only, Mendel reached the conclusion or he could manage to tell us that only one character among these 2 is dominant. That means they appear in the first generation. So you know that just forget about the pea plant. We can now move on to the human beings. You know that in the human beings, how many chromosomes are there? In every cell, how many chromosomes are there? 23 pairs of chromosomes, that is 46. Clear? And you should understand how these pairs, pairs, likewise only. Yes, this is one pair. Yes, assume. And this is the another pair. Just imagine this is 3 pairs. Likewise, 23 pairs are there. 23 pairs of chromosomes are seen in every cell. And these chromosomes, you could see the DNA. In chromosomes, DNA is a double helix model. In the chromosome, you can see the DNA. And on the DNA, we could see the genes. On the DNA, we could see the C. For example, if consider us, some humans they are tall and some are dwarf. Just an example. And that every character is controlled by 2 factors. Mental call it as factors, or we can call it as what? Genes. Every character is controlled by 2 genes. Who can tell me the answer of that? Why is it so? Why for our example, our hair color, our eye color, our body height, our body color, and etc. etc. All these characters are controlled by 2 genes or 2 factors. Why is it so? Why the 2 genes are responsible? Yes, because it is coming from 2 parents, isn't it? It is coming from 2 parents. It is because of that only every character of an individual is controlled by 2 genes. See, now observe very carefully. These 2 genes, one gene is in one chromosome and another gene is in another chromosome. In the case of tallness, just imagine, one character is one chromosome, another in the another chromosome. Clear? All the characters. If in the case of color of the skin color, you can just S is single denoting. Then another one is in another chromosome. We can't see these 2 genes in one chromosome. As they are in pair, one gene is in one chromosome and another gene is in the another chromosome. Hope it is clear how the genes are located. Not in a single chromosome as it is paired. 2 chromosomes are there. So one gene in one chromosome and the opposite of that. It can be seen in the another chromosome. Clear? See what's the benefit of it. All the cells in our body has got 23 pairs of chromosomes except what? 2 cells are there. Yes, in the case of male, it is the sperm, and in the case of female, it is the ovum. Sperm and the ovum or otherwise the egg. These 2, they have only 23 chromosomes. See what happened. At the time of gamete formation, that only one cell will move to the sperm. From these 2, one pair, any of this move to the only one. Only one will move towards the sperm. Similarly, one will move towards the ovum. The pairs will not move together. From this pair, only one will move towards the zygote. Towards the gamete. That is because of that they have only 23 chromosomes instead of what? 23 pairs that are seen in all the cells. Clear? Hope it is clear. Then we can move on to that. So these chromosomes are always seen in pairs. So every character is controlled by 2 genes or 2 factors. That means, and how they are situated. One here in one chromosome, another factor or another gene in the another chromosome. At the time of gamete formation, we know that gamete has got only 23 in the case of human. Now pairs are there. You know how it happened at the time of gamete formation. One of these, any chromosome can move to the gamete. Anyone among the pair, any one chromosome, they move towards the sperm or the ovum. Clear? That is to maintain. Why these the gametes, they have only 23? Yes, we already discussed about it so many times. To maintain the number of the chromosome in the newly formed zygote, because 23 from father, 23 from mother, then the zygote will have how many chromosomes? Yes, 46. To maintain the stability of the number of chromosomes, we could see that gamete is having only haploid or 23 chromosomes. So now we can move on to the, just forget about the human beings. Now we can do the monohybrid cross once again. As this is very, very important, it is because of that I'm repeating, and I would like to do 2 more examples. Clear? To make you understand it better. The last class, after explaining the one character, I asked you to do the workout 2 more examples. I don't know how many of you have done it. If you have any more doubt regarding it, now just watch this class once again. Clear? Okay. So this here, first he selected what? Tall and dwarf. Listen very carefully. Tall and the dwarf. You know, he's like pure tall plant. You know what is pure tall plant? He selected pure tall. You know that as tallness is the dominant one, 2 conditions are there for becoming a pea plant to be tall. 2 conditions are there. First one, the 2 genes are there should be of same. That is both capital T and capital T. Second condition, who can tell me? Yes, 1 capital T and 1 small t. So this alternative form of the gene is known as what? Allele. This is the alternative form, capital T, small t, and this alternative form of the gene is known as the allele. It is known down. Alternative form of the gene is known as the allele, because 1 capital T, this alternative form is the small t. Clear? Yes. So if the both are same, we can call it as what? Homozygous. Another term, just note down. If both are same, it is homo means what? Same. Then what about different? It is the heterozygous. If both are same, it is the homozygous. If both are different, it is the heterozygous. So what is allele? It's the alternative form of the gene. There's another form of the gene. Here capital T is the gene, or another form is what? Small t. If both the genes are same, we can call it as what? Homozygous. If they are different, we can call it as what? Heterozygous. Clear? Yes. So why here pure tall means what? Both the T should be the same. Otherwise, the result will not be sufficient enough for him to find out the correct answer for his question. Clear? So these 2, it should be pure tall. So you may have doubt how he selected pure tall. So after doing a lot of experiments and after he selected the genetical makeup and all, he could manage to find out which one is the tall plant. Clear? Yes, pure tall. That is very, very important. In first section, he should select the pure tall plant. And you know that pure tall plant, the capital T, pure tall. And what about dwarf? Very simple, small t, small t. So you know that this plant, tall plant is having these 2, they remain together for a long time. As a pea plant, it has got this tallness, 2 what genes remain together. But at the time of gamete formation, hope you remember the law that you had already studied. At the time of gamete formation, what happens? They separate, and what will happen? Either of these, you had already seen, 1 T will move to the zygote and separate. Separation is there, then another, that is the T. Any of this T, you know the gamete formation, T here T. So this T will move here and this small t will move here. So this is the first F1 generation or first female generation. So these are the 2 genes remain together at the time of gamete formation. You know they separate. They separate. That means 2 T's are formed. And here also you can see 2 T's are formed here also. And anyone can fuse with this. Okay, anyone can fuse with this at the time of gamete formation. You could see that, if they combine together, both conditions are same. Here also capital T, small t, and here also if they join together also capital T, small t. No change at all. So that's very important. At the time of gamete formation, even though these 2 genes, they remain together, at the time of gamete formation, they separate and segregate. That's very, very important. Law of segregation. We discussed about 2 laws in the last class. Law of dominance and law of segregation. In the law of segregation, I mentioned that, or the definition indicates that even though these gametes, the genes, they remain together, but at the time of gamete formation, you could see that separate and segregate, and they combines with another gene. And you know that the first generation, F1 generation, or they have got what? It is the Mendel could see that all the plants are tall. And he got really confused because he had crossed tall and dwarfness. And he called this tall as what? Dominant. Another important thing. Because what happened? It expressed. So he called it as what? Dominant. And another character, that is the small t or the dwarfness, he called it as what? Recessive. That is it. He called it as tall as the dominant because it expressed in the first generation or first filial generation. See, so he called it as dominant. Another one, it is the dwarf because it is not expressed. So he called it as what? Recessive. Clear about the first thing? You know, capital T, listen carefully, small t, and you know that two gametes are formed. Clear? Then capital T, small t. Here also same thing, capital T, small t. So it is the tallness. Clear? So he wanted to find out what happened to this dwarf because already dwarfness is there. And what did he do? He allowed the plant to do the self-pollination. Here he did what type of pollination? Cross. Because two plants are involved in what? Cross-pollination. Two plants are involved. Here only single parent. In the second generation, he liked to do the experiment with only single plant. That is this plant, their genotype is what? Expressive character is known as the phenotype. Expressive character is known as the phenotype, and genetical makeup, it is known as the genotype. Expressive character is known as the phenotype. Here what is the phenotypic character? Tallness. Because the plants that were produced after the crossing, all the plants were tall. So that is the phenotype. And what about the genotype? Genetical makeup. When he observed the genetical makeup, he could see the small t also there. So he wanted to find out, will it appear in the next generation or not? So you know that both parents are the same, father and the mother. Clear? So we can just do this by form of a Punnett square. Otherwise also we can do it. See, capital T, small t. Both parents are the same. Clear? Both parents are the same. So here capital T, capital T, capital T, small t, capital small t, capital T, and small t, small t. Clear? These two, T, capital T, small t, capital T, small t, capital T, or both way you can write small t, small t. Clear? So here also we can do, we can just separate it. T. Okay. Then this T you can fuse with this T. Then what we will get? Capital T, capital T. Here capital T, small t. Then this T is balanced here. Small t, capital T, and this can be a fuse small t, small t. Clear? Same result here also. Capital T, capital T. How many are there? Only one. Capital T, how many? One. Then what about capital T, small t? How many are there? See, two. Capital T, small t, two. And what about small t? In both cases, you can see one. And what about the normal ratio? And among these, how many plants are tall? This one. What about this? Yes, this also tall. What about this? This also tall. So how many tall are there? 3. And what about the dwarfness? Only 1. So what's the ratio here? 3:1 ratio. Phenotypic ratio, very important, expressive only. That is 3:1 phenotypic ratio. Then what about the genotypic? When he observed the genetical makeup or the genotype, he could see that only one pure tall. Very clearly you can see only one pure tall and two are the intermediate. Both are there. Even though the size of the plant is tall, but the genetical makeup, one capital T and one, both are there. There's an intermediate 2. What about the dwarfness? Only 1. So the genotypic ratio, it is 1:2:1. Very, very important. Phenotypic ratio, it is 3:1. And genotypic ratio, it is 1:2:1. 1 is here. Here, this is this 2. We could just mark it AB. This is 2, then this is the C. See, 1:2:1. Clear? 1:2:1. So, that is the genotypic ratio of the gene. Then the 3:1, it is the phenotypic ratio. Hope it is clear to do the cross. Then we can do that in one more. Yes, we can do one more experiment, sorry, one more cross here. That is, and why it is known as monohybrid cross? Because he selected one pair of contrasting characters. Then you may have doubt that that plant doesn't show any other characters. Of course, but Mendel concentrated only, by doing this experiment, Mendel concentrated only in one pair of characters. Of course, as I told you, 7 pairs of characters are there. So the plant may show different around, maybe seeds are there, pod color, flower color, differences are there. But he concentrated only on first experiment. Afterwards, definitely he had done experiments with all other characters that you are not supposed to do this year. But he had, and one more cross you are supposed to do, dihybrid, because he selected 2 pairs of contrasting characters. The next class I will discuss about it. So now we can do one more experiment regarding monohybrid cross. That is the, as I told you, the color of the flower, that is the purple, and another is the white. And purple color he denoted as both capital W. Then of course it will be, just try to say along with me, then you will understand it better. Okay. So what about the gametes? Capital W, and here gametes, small w. Next one is small w. Then first generation, yes, he crosses this W. Then what was the result? Capital W, small w. Here also, what is will be the result? Capital W, small w. Then that is which generation? F1 generation. F1 generation. Don't get confused. As you see W, don't think it is white. Mendel had given that W for the purple. So don't think that W indicates white. No, capital W doesn't indicate white. Capital W indicate what? Purple only. Very, very, please note that. And here, F1 generation, all the plants were which color? Yes, purple color. So what happened to this one? Again, Mendel want to find out what happened. So he allowed the plants to do the self-pollination. So we can just indicate through the Punnett square. Yes, capital W, we can separate this, okay, capital W, small w. Here also, both parents are the same, capital W, small w, small w, capital W, small w. Yes. Tell me. Yes. This. Yes. So, observe it. How many pure purple flowers are there? 1, 2, and 3. What about white? Only 1. That is the expressive ratio or the phenotypic ratio. That is the 3:1. Then what about the genotypic ratio? Here only 1. Pure purple, only 1. And these 2, it is the intermediate. 1, see, capital W, only 1. Capital W, small w, 2. Small w, 1. So 1:2:1. Clear? This is the genotypic ratio. So for the exam point of view, you may be asked to write the ratio of in the monohybrid cross between the purple or white or tall or dwarf. Right, in the F2 generation, what's the ratio of pure tall to dwarf? Pure tall to dwarf. Then how will you write? 1:1. Here only one pure tall. Of course, pure dwarf. Then pure tall to intermediate. 1:2. Intermediate to dwarf. 2:1. Hope it is clear. So in different manner, these questions can be asked for the examination. Clear? So I hope it is clear how to cross this. Purple, capital W, white, small w. Then gametes, they separate at the time of gamete formation, that is law of segregation, and here also separate, and they combines with another factors. And in the first generation, all the plants bear purple flowers, okay, and he did the self-pollination to the F1 generation plant. Then what was the result he received or he got? That 3:1 ratio, that is the phenotypic or expressive, externally expressive character. It is known as the phenotype, 3:1. But when we observe the genetical makeup, and he got the result that only one pure purple and two intermediate, even though that appears to be purple in color flower, but the genotype is what combination of these two, that is intermediate two are there likewise, and the last one, it is the dwarf, it is the white color flower. Clear? So you should understand phenotype and genotype, very very important terms. Phenotype is the expressive one, and genotype, it is the internal or genetic. Gene can be seen inside, isn't it? So that is the genetical makeup. So one more example we can do it. Okay, one more example. Last one. So please take a paper. Please do it along with me. This one. Then only you will understand. Now you may feel that everything is clear to you. But while you work out only, you will understand. You can again feel what confusion. So please take the paper and do along with me. That is we can see the seed, that is the round. That is the dominant one. And next one is the wrinkled. Okay. So round can be indicated, capital R, capital R. Then this will be, please write along with me, smaller, smaller. So how can we separate the gametes? Capital R. Yes, capital R. This one. Smaller. Smaller. Then combine these two. Capital R. Smaller. This one. Capital R. Smaller. This is which generation? F1 generation. And all the plants were bear which seeds? Round seeds. Now he did the self-pollination. So we can separate these gametes, capital R, smaller. Here also capital R, smaller. Clear? And we can do by means of Punnett square here. Capital R, smaller. Capital R, smaller. So try to do it. Yes. Capital R, capital R. Here capital R, smaller. Capital R, smaller and smaller, smaller. Hope it is clear. So how many pure round seeds are there? Yes, only one. Clear? Then what about the intermediate? 2. Okay. Then what about this? 1. So the ratio. How many round seeds plants are there? 3. And what about wrinkled? Only 1. That is the which ratio? Phenotypic ratio, or otherwise we can call it as what? Monohybrid cross ratio. Clear? Phenotypic ratio or monohybrid cross ratio. Then what about the genotypic? Here only one, this 2 and this 1. 1:2, 2:1. That is the genotypic ratio. Hope it is clear, my dear children. If it is clear only, I can move on to the next session, that is dihybrid cross. Clear? Okay, so a small section also I would like to include here. It is about, then you may wonder how these genes, genes, they control the character of an individual. And why is this so? You know that cell has got what? This is the cell. This is the cell. Cell has got what? Nucleus. Inside the nucleus, we could see what? Chromosomes. Of course, in the paired state. Clear? Okay. So, in this nucleus, this is the cell. This is the nucleus. And these are the chromosomes. So, in the chromosomes, we could see the DNA. Inside the chromosomes, we could see the DNA. So, DNA is often called as information source for making proteins. DNA are the information source for making protein. Information source for making proteins. DNA, they are the information source for making proteins. And DNA is unique. That is, my DNA is different from your DNA, even though the twins, of course, in the case of some rare cases, like identical twins only, they have, sometimes they may have similar DNA. Sometimes only. In all other cases, DNA is specific, that is, and unique. It is particular to particular individual. So, this chromosome, they have DNA. As we all know, DNA shape, double helix model shape. So, then DNA is a section of DNA. A section of DNA. Section of DNA that provides information for a single protein, that is known as what? It is the gene. What's a gene? It's a section of DNA that provides information for a single protein. That one mark question you can expect. What's a gene? A section of DNA that provide information for a single protein. For example, in the case of height, a single gene can provide information for what? Height only. So, that in the case of, we can again move on to the plants. Last chapter we discussed about the hormones. So, plant hormones are there that can provide height to the plant. And if plant hormone is produced in an efficient manner, the plant will be tall. If it is not produced in an efficient manner, plant will be dwarf. So, efficiency. The efficiency of producing the hormone, it lies in the gene. If the gene is efficient enough to produce a particular protein, if gene that controls the height, if gene is efficient enough to produce a protein that triggers or that help or that increases the efficiency of producing the growth hormone, the plant will be tall. If that protein is not efficient enough, of this gene is not efficient enough to produce the protein that help in the plant growth, what will happen? The plant will be dwarf. That is it. So, the gene is a section of DNA that provide information for for producing a single protein. So, this protein, different proteins are there. This protein, they control different characters. Each gene control specific characters. Tallness of the gene control only tallness. Then color of the gene only control color. And if that gene that is efficient to make a protein that can make an individual color, means white, that means that that individual will be white. Or if that gene is not efficient enough to produce the protein, then that person will be dark. And the height, of course, and if that gene is efficient enough to support the production of growth hormone. In the case of human beings, last chapter you studied about growth hormone. And if the gene is efficient enough to produce the protein that help to produce what? That help in the production of growth hormone. Gene produces proteins, specific proteins, and that protein is efficient enough to support the production of growth hormone, the individual will be tall. But if that protein has some problem in helping that, or if that gene is not efficient enough to support the growth hormone, definitely that individual will be dwarf. So here the DNA plays a very important role because DNA are the information source for making proteins. DNA are seen in the chromosomes. Inside the DNA we can see the genes, and you know the genes, they are seen as paired. One character can be seen in one chromosome, another one in the next chromosome, and at the time of gamete formation only from one pair only one chromosome move towards the gamete. They don't move together. From one pair, only one chromosome. So how many pairs are there? 23 pairs are there. So 23 chromosomes will move towards the gamete, and every chromosome has got DNA. Every DNA has got different genes for controlling each and every characteristics of every organisms. Clear? Yes. So now again, please try to work out some monohybrid cross. Then make it clear. When we meet next class, I will explain about dihybrid cross, children. So please be serious about your studies also. So your exams, I think approach on 28th. So be ready for it. Okay, my dear children. Until we meet in the next class, it's bye from me. See you. Take care. Be safe. God bless you, dears. Bye.